So you should have printed out the two brain dissection worksheets. One is going to have a fill in the blank at the top and then some charts at the bottom, and then the other one is going to be some pictures of the brain. You're also going to need your colored pencils because we will be labeling and coloring structures of the brain. So let's begin with the fill in the blank at the top. There are 4 sections to your brain. 4 main areas. That part that is colored purple is the largest part, is the cerebrum. The second largest part is that's highlighted light green, that's the cerebellum. Then we have the brainstem, which is orange. And finally, the inside, which I've highlighted light blue, is the diencephalon. So 3 of the main regions you can see externally, 1 you have to go deep. The diencephalon is actually made of the thalamus, the hypothalamus, and the epithalamus, or the pineal or pineal gland. So let's look at the brainstem. Your brainstem actually is composed of 3 structures. The structure that joins with the spinal cord, which is the link between the spinal cord and the brain, is called the medulla, sometimes medulla oblongata. Next, going towards the brain, is the pons, sometimes the pons varioli. And then last is the midbrain. And in the midbrain, you find the corpora quadrigemina, and sometimes you refer to the pineal gland being somewhat in the midbrain. So that should answer number 2 on your sheet. Looking at the outer surface, the cerebral cortex, the outer surface of the cortex, you see that there are hills and valleys. The hills or the ridges are called gyri. A single one would be a gyrus. And then the valleys or the grooves are sulci or a sulcus. You may remember sulcus from a groove on a bone. So sulcus would be the grooves. And these ridges, these hills, and these valleys give us more surface area so that we can have more neuron networks and store more memories and information in our brain. And then you'll notice too that on the second picture that the 2 hemispheres of the cerebrum are divided by a very deep groove. This groove is called a fissure. That is actually the longitudinal fissure. So deeper grooves are called fissures. The cerebrum is divided into 5 lobes. If you look on the picture, you can see the 5 lobes that are named after the brains, the bones of the skull that correspond with these brain structures. Remember the frontal bone? So there's a frontal lobe. Parietal bones, parietal lobe. Occipital bone at the base of the skull, occipital lobe. And then the temporal bone, which was up over the ear, is the temporal lobe. So we name these 4 outer lobes by the bone that covers them or protects them. Now if you pull down the temporal lobe, you can see that there is another lobe that's deep, and it is called the insula. So on your sheet, the insula is buried deep, but the remaining 4 are named for the cranial bones. Insula. It's in there. So there are 5 lobes. The one that's deep is the insula. Now let's talk about the meninges. Surrounding your brain, there are membranes. If we go superficial to deep, there are 3. These membranes are called meninges. The outer one is very tough, so it's called the dura mater. In Latin, that means tough mother, so it protects like a mother. It's very tough. The next layer has a lot of blood vessels associated with it, and so it has these fibers, and it looks like a spider web. So someone named this middle meninge the arachnoid mater, the spider mother. And then the one that's adherent to the brain tissue is called the pia mater. So you have the dura mater, the tough meninge, the arachnoid mater, the one that's more associated with the blood vessels, and the pia mater, which is adherent to the brain surface. When we dissect the brain later, you'll see me cut off the dura mater, and it is very, very tough. It helps to protect the brain. So you have the cranial, the skull bones, the cranial bones that help protect, and then you have this meninge that also helps protect. Now, inside your brain, there are ventricles or spaces, and inside the spaces or ventricles are networks of blood vessels called choroid plexuses. And so in the plexuses, we make cerebral spinal fluid. This is another way that we protect the brain because we're making a blood-brain barrier. Instead of the blood vessels attaching directly to brain tissue, they are found in these ventricles, and it's almost like we filter the blood of nutrients and oxygen to make cerebral spinal fluid, and then the cerebral spinal fluid bathes the nervous tissue of the brain and the spinal cord. So it's made in ventricles, and you can see on this picture a lateral ventricle with a choroid plexus in it. That's on either in each hemisphere, so there are two lateral ones, and then you'll see the third one down right above the pituitary gland, and then the fourth ventricle is above the cerebellum. I'll point these out when we get to the brain dissection as well. So on your sheet it says the cerebral spinal fluid is formed by the choroid plexus in the ventricles of the brain. The fluid circulation is from the lateral ventricles to the third ventricle to the fourth ventricle and down the subarachnoid, down the spinal cord and up through the subarachnoid space, bathing the neural tissue, protecting it from anything that might have directly attacked it in the brain, in the blood, I'm sorry. This is just another picture that shows those ventricles and those choroid plexuses that make cerebral spinal fluid. Now I know when I videoed the real brain dissection, I pointed out the third ventricle and a choroid plexus, and in my review of the video, I realized that I said the choroid plexus is made spinal cord, and of course I misspoke. It makes cerebral spinal fluid, the choroid plexuses. So let's look at the picture on your lab sheet. This is the second page of the lab sheet. Let's label some of the structures that we've already talked about. This upper area, the largest area of the brain, is called the cerebrum. Cerebrum is number 5. So on your sheet, on the front of your sheet in the box where it says cerebrum, put a number 5, and then later you're going to go back and you're going to color code that whole cerebrum one color and color the word cerebrum that same color. So let's label the second largest structure. The second largest structure is the cerebellum, and it has a number 6 going to it. So on your chart, cerebellum would be number 6, and later you will color code the cerebellum. This portion of the brain actually has 3 parts. It's known as the brainstem. Brainstem, so on your chart, brainstem would be number 30. Brainstem would be number 30. So record number 30, and later after the video you can go back and color code it. So cerebrum, cerebellum, brainstem. There's one more region of the brain, but it's found internally, and it is the diencephalon. Number 31 is going to the diencephalon. So the diencephalon is 31. I often remember diencephalon because cephala means head or towards the head, and I figure you would die if you went inside to see the diencephalon of your head. This is a corny way to remember it, but diencephalon. Now while we're on this view, we're going to go ahead and label this gland that hangs from the ventral surface of the brain. It's the most protected gland in the body because it rides in the sella turcica of the sphenoid bone. It is the pituitary gland. So the pituitary gland is number 8. On your worksheet, it's in the chart on the right hand side that says ventral exterior view, pituitary. Go ahead and record that as number 8. So let's look at some of the deeper sulci or grooves or fissures that divide the cerebrum into lobes. This front lobe is known as the frontal lobe. it corresponds with the frontal bones of the skull. So on your second picture of the external view of the brain, you'll see a number 1 that I've drawn over the lobe. So on your sheet, frontal lobe, you should label it number 1. This lobe corresponds to the bone that makes the top and sides of the skull. So this would be the parietal lobe. This lobe at the base of the brain matches the bones at the base of the skull, so this would be the occipital bone, or occipital lobe. And then this one matches the bone that's on the lower sides of the skull, the temporal bones, so this is the temporal lobe of the brain. So on your sheet, you should have frontal lobe 1, parietal lobes 2, temporal lobes 4, occipital lobes 3. So there are some definite structures that are going to divide these lobes, but before we look at those, let's go ahead and label that cerebellum again. Even though we've already recorded it on our box, we can go ahead and label it again on this picture. Now here is the first deep sulcus that I want to point out. This one separates the frontal and the parietal lobe. It is called the central sulcus. So central sulcus would be number 24. Another sulcus that's pretty prominent that divides lobes is this one. This one divides the temporal from the frontal and a little bit from the parietal. This one is on the side of the brain, so this one is called the lateral cerebral sulcus. Now there are some other prominent sulci, but these are the two we're going to focus on, and we're going to focus on that deep fissure that divides the cerebrum into two hemispheres. Do you remember what I called that? It's the longitudinal fissure. And so you'll see the skull, and it has the brain sitting in it, and I have a number 7 going to the longitudinal fissure. So now on your dorsal exterior view chart, you should have, starting from the top with cerebrum, 5, 6, 31, 30, 7, 24, 23, 1, 2, 4, and 3. So in the brain dissection, you will see me actually remove the meninges. So if you were in a traditional lab, you would have to take scissors and very carefully remove that dura mater and a little bit of the arachnoid mater. So if we remove the meninges, then we can see some other structures of the brain. If I hold the brain in my hands and I pull gently back on the cerebellum, I can see a structure called the corpora quadrigemina. It actually is made of 4 parts, 2 superior colliculi and 2 inferior colliculi. It almost looks like a little booty mooning you there. Superior colliculi then would be number 9 on your worksheet, and inferior colliculus or colliculi is plural would be number 11. And collectively or combined, they're called the corpora quadrigemina because there are 4 parts. So corpora quadrigemina would be number 10. This is just another picture showing the superior and inferior colliculus and the corpora quadrigemina. Now if I were to take a probe or my thumb and pull down on the corpora quadrigemina, you can see that another little structure pops up, and that is the pineal or pineal gland. I've heard it pronounced both ways. This is an endocrine gland. It is the one that releases melatonin and sets up your biological clock. So on your sheet you see pineal still in that dorsal external view, and it is number 12. It is number 12 on the picture. It is number 12 on the picture. Later we're going to label it, but it is number 12 on the picture. So let's go to a ventral view of the brain, and let's learn some structures that you will identify later on the dissection. We're going to start at the bottom of this picture with the spinal cord. So this is the spinal cord. What number is going to spinal cord? 16. Then we come to the brain stem. The first area of the brain stem is called the medulla oblongata, or medulla for short, and that is number 15 on your picture. The next part of the brain stem is called the pons varioli, or just pons, and that is number 14 on your picture. The next part of, or the last part of the brain stem is called the midbrain, and so the midbrain is number 25 on your picture. Then we see a small mound of tissue. This is called the mammillary body. Right above the mammillary body, we see a hole that once was the stalk that held the pituitary gland on. It's called the infundibulum, and so that would be number 28 on your picture. Now, on your picture, it looks more like a stalk. On my picture on the screen, it looks more like a hole. But if you can imagine that the pituitary gland was once held on are held to the ventral surface by a stalk, and now all we see is a remnant, a hole. Above the infundibulum, we see the crossing of the optic nerves. That's called the optic chiasma, and it's number 27. Above the optic chiasma, you see the olfactory tract. It's number 26, and the olfactory lobe, which is number 29. So now we have labeled several of the, or all of the ventral exterior view. Starting with pituitary is 8, olfactory bulb is 29, olfactory tract 26, optic chiasma 27, infundibulum 28, mammillary body 13, brainstem, the midbrain is 25, the pons varioli 14, the medulla oblongata 15. Now, something I want to show you very quickly is what I was showing you earlier, that there are ventricles in the brain, 4 of them to be exact, and inside those ventricles is a mass of blood vessels called the choroid plexus, and those choroid plexuses actually produce cerebral spinal fluid, kind of make the blood-brain barrier. So the 2 laterals are on either side of that corpus callosum, or that structure that separates the 2 hemispheres of the brain. So you have 2 lateral ones. Above the intermediate, or that round body, is the 3rd ventricle, and then underneath the cerebellum is the 4th ventricle. So the 3rd ventricle is going to be number 21, and the 4th ventricle is going to be 17 on that lowest picture on your worksheet. Now, I went ahead and labeled 9, 10, and 11. We've actually already put those numbers on our worksheet. Remember that 9 is the superior colliculus, 11 is an inferior colliculus, and collectively they make up the corpora quadrigemina. This, you should remember, is the spinal cord. This is the medulla. This is the pons. And so that would be 16, 15, and 14 on that picture on the bottom of the page. This, of course, is the cerebellum. And inside the cerebellum, we see the tree of life, or the arbor vitae. So this is the cerebellum, and the arbor vitae then would be number 22. Here again, we've labeled the corporal quadrigemina and the superior and inferior colliculi. Remember, above the corporal quadrigemina, when we pulled back the cerebellum, we saw the little pineal or pineal gland emerge, and so that is number 12 on your picture. The white matter that holds the 2 hemispheres together is called the corpora callosum, and it is number 18 on your picture. As the corpus callosum bends around, it is called the fornix. The fornix. The mass, round mass, that's very distinctive when you open or do a mid-sagittal section on the brain. That's where the thalamus is, but it's called the intermediate mass, and that is number 20. This would be the pituitary gland. And you can see right above it there is a stalk leading up. That would have been the infundibulum. And those are the structures of a mid-sagittal section. So on mid-sagittal view, you should have 16 spinal cord, 15 medulla oblongata, 14 pons varioli, fourth ventricle 17, corpus callosum 18, fornix 19, intermediate mass 20, third ventricle 21, arbor vitae 22. So now that you have your sheet filled out, before you submit it, you will color code it, remembering to color the word, the color, unique color pattern, and the structure, the unique colored pattern. You can use solid red, outlined red, X'd red, dotted red, swirly red, squared red, striped red. So you can use a unique pattern for each one of your structures. Make sure now that you watch the dissection videos that I prepared for you. Also on My Labs Plus, there is a brain dissection there as well.